So, when should you never take a compliment? Well, if you're bacteria. The immune system is an amazingly complex series of proteins and cells, all operating along the same goal of defending you against any invading organism, including bacteria, viruses, protozoan, parasites, you name it. They're your first, second, and last line of defense against any invasion. Now, as you involve more and more cells and proteins and systems into the defense against microorganisms, the increasing complexity means that it's going to take more time. And so your body is built in a way in which you can have both immediate responses to infection and then also more delayed responses that tend to be a little bit smarter. The general theme here we're going to talk about is we're going to start with the simple immediate response that doesn't require a lot of complex interactions with cells, all the way up to the interaction of B and T cells involved in the actual specific defense against a specific microorganism. So one of the first steps in the immediate response to infection is the complement system. Now, complement is your first weapon and is a series of proteases that are produced by the liver and circulate free throughout your blood system. Now, these proteins, because they're proteases, circulate initially as inactive enzymes, and then in the presence of an infection can trigger a series of proteolytic reactions that we call complement activation. Now, the complement system, although it is a series of proteins, actually involves up to 30 different enzymes. And immunologists have used a naming classification scheme where the letter C denotes it as a complement protein, and then the number that follows it to denote when it was discovered in its sequence or its sequence in activation. However, at the end of the day, no matter how the cascade is started or what proteins are being involved, the big money is on the complement C3. C3 is a complement protein that circulates free within the blood, just like all of the other proteins. And when it is activated, complement C3 is cleaved into two fragments. One is C3A and the other is C3B. Each one of these fragments has a very important role in the defense against microorganisms. C3b binds to the pathogen surface in a process that we call complement fixation. This complement fixation then tags the microorganism for destruction by any other different part of the immune system, either by direct phagocytosis or formation of protein complexes that can destroy the cell wall. C3a also has an important role as it floats away from the site of infection and will act as a chemoattractant to bring other warriors to the defense of the realm. Complement activation can occur through three separate pathways. There's the alternate pathway, the lectin pathway, and the classic pathway. We're going to talk mostly about the alternate pathway in this talk, because the classic pathway requires antibody binding, either using a specific antibody or by C-reactive protein on the pathogen surface in order to activate the complement cascade. We will touch on that later in this series on immunology, but today I want to focus specifically on the alternate complement pathway. The alternate pathway is the innate pathway. It doesn't require any feedback from any other system. It's just simply the presence of a pathogen is sufficient to activate this pathway. So it is automatic and it's very rapid. The starting reaction for the alternate pathway is through the normal turnover of C3 to inactive C3 or IC3. Now, this is a normal ongoing degradation of C3 that occurs regardless of whether or not there's an infection going on or not. But when you get to a site of infection where there's some inflammation, where there's irritation of the system, and activation of other complement or coagulation or inflammatory mediators, the turnover of C3 to IC3 increases. IC3 then binds to another blood-bound protein called factor B. Now, this complex is then activated further by another factor called factor D, which then creates the active enzyme IC3BP. Now, this is a protease that is able to cleave C3 into its components, C3A and C3B. So you can see how this accelerates the identification and tagging of pathogens by increasing the local concentration of C3b, as well as acting as a chemoattractant by releasing more C3a. The IC3BB complex is also able to bind to the pathogen itself, and right on its surface it then can generate a local feed-forward system where the enzyme sits on the microorganism, it finds C3, it cleaves it, it forms C3b, and that C3b then binds immediately onto the pathogen because it's right there. This theme of a feed-forward mechanism is very common in these types of systems that are related to the defense against pathogens. And so feed-forward mechanisms will become common, and you'll see many examples of them through the course of this series. But to suffice it to say, the activation of complement occurs not only naturally just through its own natural breakdown, but then also can then be more specific by forming a protease that can then bind specifically to a pathogen and then accelerate the identification and complement fixation on that microorganism without any input from any other part of the system. That being said, no man is an island, and you do need to have some help if you're going to effectively deal with any pathogen in your body. And one of the first effector cells that we're going to encounter is the macrophage. And the macrophage is one of the first effector cells in the innate immune system and is a mature monocyte cell that gets converted from a monocyte into a very long-living phagocytic cell. Macrophages are a bit of the garbage collectors of the body. They're non-specific. They'll bind to and eat up just about anything that they can encounter that seems foreign to it. It's very non-specific, and so it phagocytizes anything it can pretty well get its hands on. This phagocytosis, though, becomes much more efficient when it encounters a pathogen that's covered with C3b. Because macrophages have a 3CB specific binding protein on their surface that helps it identify and then rapidly aggregate and phagocytize complement-coated organisms. Now that's all well and good because it's nice to have some help from the garbage collectors, but C3b does not rely specifically or exclusively on macrophages to help it do its dirty work. The alternate complement cascade actually has this really neat set of steps that it can go through to actually kill a cell without involving any cellular element of the immune system. IC3BB can bind to another fragment of C3b, and this forms what's called a C5 convertase. This C5 convertase convert C5 to C5a and C5b. Now C5b then initiates a membrane attack complex that pokes a hole in the bacteria and actually any cell that it happens to be bound to, so bacteria and eukaryotes. The sequence of events is fairly straightforward. Once you have C5b, it inserts into the cell membrane of the pathogen using both C6 and C7. C8 then binds, and once you have this complex, it initiates the polymerization of C9 to form what's called the membrane attack complex, which then perforates the cell and forms a transmembrane pore, which allows the cell just to basically explode along its osmotic gradients. So today we learned about the immediate response to an infection. This is the system that is primarily protein developed and utilizes only one cellular element, the macrophage, which is the garbage collector of the system. The complement system is all proteins that float free in the body that allow it to identify, tag, and then in certain cases actually destroy invading pathogens. Next time we're going to talk about the innate immunity and the induced response to an infection.